Good evening. This is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the beautiful late fall inland Pacific Northwest of the USA. Today is the 4th day of December and the year is 2021. Not too much of this year left, and that's just fine with me. We are actually at the final episode of a year-long journey into our discussion of the dialectical event ontology of aging. So I'm labeling this La Lezione Finale, the final lecture on the topic. It's going to be a long one, even though I've tried to summarize and carry out a synopsis of over 50, maybe as much as 60 hours of lectures that have gone through ever since November of last year. And what I want to do in the end is give you my discrete understanding of what I'm now calling the sickness unto death, aka aging. And of course, a nod to Soren Kierkegaard, who was talking about the sickness unto death, describing despair. And because I'm a biochemist, I'm taking that topic name and the title of a book he wrote, and I'm converting it into something about physiology and biochemistry rather than philosophy and psychology. But it basically gets us to the same point, I think. So let's get started. Again, this is Dr. Dan Guerra, and you have landed into Authentic Biochemistry, so I hope that you enjoy it. I'm going to, as I normally do, put a visual of myself giving a lecture somewhere around the screen. I apologize, sometimes I have to move it, and I know that's distracting, but I don't have a person that can help me manipulate that, so I do it all right here with the keyboard. So pretty amazing, right? I do the content, deliver the lecture, and I'm doing all this fantastic presentation. So I know you're going to be at least moderately impressed with it, okay? Okay, so this is actually a picture I took when I was in Canyonlands. I don't remember, maybe 2-3 years ago. That's near Moab, Utah. One of my favorite locations. Okay, so the photographs that I use, for the most part, are ones that I've taken. If I don't, otherwise suggest that they came from somewhere else. Okay, so I don't have to pay for this, right? Isn't that nice? It is. Now, doing this as a means to disambiguate the pleiotropic biochemical and physiological phenomena of human aging unto death. Because, and to get to that point, it is necessary to explore the molecular events of metabolic pathways within changing cellular networks over the natural lifespan. That's why it's taken me a year to put this together, delving into the primary research in peer-reviewed literature. Okay. I know that those of you that have watched this regularly know all about this. I'm just making sure that if you haven't listened to me before, you know that that's where we get all the information. Not from popular press, not from newspapers, not from other podcasts, from me reading the literature, synthesizing it, and then presenting it to you. So I use a dialectical approach because it allows what I'm calling an either-or means of analysis. So we're doing a study of the agent human through the lifespan, and it's going to draw upon an event rather than a substance ontology of living systems, the living system, that being the human being. Because of the temporal nature, because it's an event of living unto death, and that is the final stage of living is death, the exposed mechanism involves a trigonal planar dimensionality of the lived experience that requires three aspects of interaction: than a temporal framework. Genomics, environment, and what I call the neuroimmune epigenomics. Again, we've talked a lot about this in the past, but I'm summarizing. So hopefully, if this isn't review, this won't be something that you haven't at least been exposed to before. So I'm calling this trigonal planar structure here a dia-eventome. And I'm saying that biological networks are processes becoming eventual in nature as opposed to substantial. By that I mean, rather than understanding things by substances such as enzymes or lipids or carbohydrates or nucleic acids or microglia or Th2 cells, those are all involved in this discussion, but they're not being described according to their substance, but according to their event ontology, how they actually function dynamically. Okay. So this event ontology reaches across the biological network of genetic and environmental interaction, and it will be articulated and synthesized through epigenetic modifications of the classical and acquired immune system, thus creating a responsive, plastic, and elastic memory field capable of metabolic scanning, focus, accession, mediation, learning, and reacting to stimuli through time. Now, the environment is the existing individual, because aging is an individual process. Although there are many principles we can gather up, and we will and we have, each individual is going to age at a different rate because this is a complex system that involves hundreds of billions of events happening virtually simultaneously. So you have to talk about an existing individual agentically that is making decisions, interacting through the events obtained across his inherited genome, the changing environment in which he resides and moves through, and the receptive architectonic foundational structure of the neuroimmunoepigenome, which itself gets written, read, erased, and then can be rewritten, read, and erased in different ways through the lifespan. So, specifically and ideally, the neuroimmune epigenome is the following. It's a biological phenomena involved in the a priori synthesis of a reception and signal transduction cascade network, RSTCN. Now, the pattern of cohering events appear as energetic and mass dynamics, much like chemical bonds. The dialectical interactions between environment and inheritance event a trigonal planar axis ontology, as I've demonstrated here. And that's via stochastic or pseudo-random epigenetic retailoring of what is essentially a biological communication system using the immunological domain to carry out the process. So what process are we talking about here? Cellular senescence. Senescence is a term we use rather than aging when we talk about living systems. Cellular senescence refers to the essentially irreversible arrest of a cell that occurs when cells experience potentially oncogenic stress. The permanence of the senescence growth arrest enforces the idea that the senescence response evolved at least in part to suppress that development of what would be a tumor cancer, potentially oncogenic stress. So the senescence arrest is considered irreversible because no known physiological stimuli can stimulate senescent cells to re-enter the cell cycle. Now notice I said physiological, that is within the normal lifespan. There are ways to alter senescent cell ontogeny, but these would be artificial. We can get into those later. Now, then I say, however, molecular biological manipulations, for example, the sequential inactivation of tumor suppressor genes, can cause senescent cells to proliferate. So this again can be done in a non-physiological sense by, say, using electrical stimuli or pharmacological stimuli. However, though, even beyond those artificial means, there can be a sequential inactivation of individual tumor suppressant genes that we describe in the cell cycle. And when that occurs, and it can occur physiologically or pathophysiologically, you can cause senescent cells to proliferate. And this can happen when you're aging, during the process of this aging. Key point. Now, there may be as yet unrecognized physiological circumstances under which the senescence growth arrest is reversible. There may be. But if they exist, we haven't been able to discover them. And we do know that all life ages, becomes morbid, and then dies. Okay. There is no evidence for any kind of living tissue dynamic event that is sustainable as an immortal system, because it's built into the very structure of that trigonal planar interaction, that the system will go through a developmental process and then ultimately generate a chronic illness known as aging that will lead to death. I hope I can convince you of that, if I haven't already, with all the lectures I've done before this. Now, regardless, the senescence arrest is stringent. It's established and maintained by, very specifically, when we look at one small window here, two major tumor suppressor pathways: the P53 P21 pathway and the P16 Inc 4A PRB pathway. And it's now recognized as a formidable barrier to malignant tumorigenesis. That's the senescence arrest. Now, consistent with that view, cells undergo senescence in response to a host of potentially oncogenic stimuli or their sequelae. In addition to arrested growth, senescent cells show widespread changes in chromatin organization, and with that, gene expression, cohering gene expression, due to the chromatin reorganization. These changes include the secretion of numerous pro-inflammatory cytokines. These are secreted glycoproteins, chemokines, similar kite, growth factors, and proteases, ending up in the extracellular matrix. And this is a feature termed the senescence-associated secretory phenotype, or SASP. And we've gone through this many, many, many times in lectures, the SASP phenotype. Okay, so more detail what we're talking about here. There's SASP involved in angiogenesis, cell proliferation, chemotherapy resistance, epithelial to mesenchymal transition, that's EMT, stem cell renewal and differentiation, sensu stricto inflammation, and also in a restorative capacity, tissue repair. So there's a myriad of activities of this SASP, and many factors that compose the SASP have numerous biological activities and therefore functions. All are highly dependent upon physiological context, what cell, what tissue, etc., and what age and what time. Those activities include stimulation of angiogenesis, stimulation and inhibition of cell proliferation, creation of a chemoresistant niche during cancer chemotherapy, stimulation of that EMT, which can then become oncogenic given the right growth factors and signaling, chronic inflammation, which can often lead to massive tissue and DNA repair, then leading to more mutations, epimutations, and alterations that can downstream lead to either programmed cell death, autophagy, or oncogenesis. So those are then result and relate to alterations in stem cell removal and renewal or differentiation, and the optimization of tissue repair during the normal lifespan. Okay. These hexagons shown here represent the SAS factors that act within and outside the senescent cell. And these are going to be bioactive molecules in a dynamic mode, right? So, cellular senescent causes. Again, we've talked about this multiple times. It was first formally described approximately 50 years ago, so not that long ago in terms of published science, when Hayflick and his colleagues showed that fibroblasts did not proliferate indefinitely in cell culture. Those cells were said to have a finite replicative lifespan and later to undergo replicative or cellular senescence, which basically is just termed replicative or cellular aging. They can no longer divide. That's the key feature of a true senescent cell. And that had been called, the number of cell divisions later on have been called the Hayflick limit. So the number of divisions that cells complete upon reaching the end of that replicating lifespan is called that. The link between the Hayflick limit and aging was for many, many decades conjectural and tenuous, although it was deeply studied. It's largely on the basis of that replicatively senescent cell phenotype appears to be associated with a degenerated, although viable, metabolically active tissue. Viable meaning it's not necrotic, it's not dead. So some 50 years ago, it was evident that most cancer cells don't have this finite replicative lifespan. They don't reach a Hayflick limit and stop. Hence, they continue to replicate or divide. And this then can lead to because of an oncogenic event, can ultimately lead to a tumorigenic response. So the idea that senescence response is tumor suppressive, although still was speculative 50 years ago, became increasingly more firmly established as some kind of cellular principle. Won't call it a law. So, paper published in 2015 in a journal called Cancers tells us the following. Cancer development can be linked to a very serious and pandemic disease known as obesity. So cancer development can be linked to obesity and sometimes spontaneous infections, which both will in fact affect cancer incidence between 14 to 20% with obesity. Now, these are very conservative estimates, to 18 to 20% when you're talking about infections from pathogens, bacteria, even parasites, fungi, and yes, also viruses. So, a common feature is inflammation. When acute inflammation is a necessary requisite for protecting the body against infection from a biological agent or non-biological pathogen, such as a virion, tissue stress and injury and genomic mutations pleiotropically will result in a dysregulated metabolism and signaling matrix pathway. That leads to an inability of the body to return to some kind of homeostasis, often promoting then what we would call chronic inflammation. Inflammation itself changes the homeostasis, as when hyperglycemia precludes dysmetabolism of, say, glucose toxicity, associated inflammation and tissue damage, such as what can occur with obesity associated type 2 diabetes, which is a major pro-inflammatory chronic disease state in many people worldwide, starting at earlier and earlier ages, thus affecting the aging response. So I'm trying to let you know that chronic inflammation predisposes the body to chronic disease state, and ultimately it can lead to either cell death or cancer. Just look at the predisposition of inflammatory bowel disease and, for example, the viruses hepatitis B and C infection, and how that maps onto colon carcinogenesis and hepatocellular carcinoma, respectively. So there's a good linkage, and there's a quite florid detail in the literature on those two topics. So more detail. Group 6A calcium independent phospholipase A2, or IPLA2 beta, which will cleave a phospholipid, a glycerophospholipid, at the SN2 position to generate a lysophospholipid and a fatty acid. Generally, that position will be held when phospholipase 2 is involved. Very long chain polyunsaturated fatty acids, and the most common one in most tissues, except neural systems, is going to be arachidonic acid. That's a 20 carbon fatty acid with 4 double bonds, and it's of the omega-6 series. All those double bonds are cis double bonds. Okay? And in fact, that may be significant in disruption of inflammatory equipoise. Just the reaction of the IPLA2 beta. So IPLA2 beta elicits physiologically important functions, which includes phospholipid remodeling, which will change membrane topodynamics, signal transduction associated with the membrane, cell proliferation, and the other side of that coin, programmed cell death or apoptosis. An inhibition of this enzyme, that one enzyme, induces apoptosis partly by preventing arachidonic acid incorporation into phospholipids. That's because there's a dynamic of release of a fatty acid and then reincorporation. Okay. Very important part of moving around fatty acids from various phospholipids in situ in the membrane with glycerophospholipids, and also with the sphingolipids, with the sphingosine backbone, which we'll get into later. So whole body deletions in a mouse model, a knockout, a double knockout, were generated by homologous recombination techniques. And it showed two major phenotypes with reduced fertility in male mice and defects in insulin secretion by the pancreatic beta cell. Now, isn't that interesting? It's a quite a far-reaching pathophenotype, right? From knocking out that one phospholipase, the non-calcium dependent, or we call it calcium independent, right? So that gives us a lot of insight into the complexity of the system. So this is caused by the production of arachidonic-induced calcium stores in the ER and an increased apoptosis in beta cell mitochondria. Beta cells, of course, secrete insulin, pancreatic cells, right? So the lack of the ability, the secretory cells, the lack of the ability of mutant mice to secrete insulin results in the observed sensitization of glucose intolerance upon feeding with a high-fat diet or treatment with beta cell toxins, streptozotocin. See how this comes together. Okay. Now, because you need this calcium alteration. Now, this is an enzyme which is calcium independent itself. It doesn't require calcium for activity, but it nevertheless can alter calcium mobility once arachidonic acid is generated from its activity. Once again, the other level, the subtle complexity and nuance of biochemistry. So upon aging to 1 to 2 years, the calcium-independent phospholipase A2 beta double knockout mouse shows a loss of bone mass associated with a decrease in body weight. And what happens in the central nervous system of these double knockout mice is you get neuroaxonal dystrophy. So yet another pleiotropic phenotypic response from the excision of this one enzyme. Right? So I say hence, IPLA2 beta deficiency at the whole body level in the mouse model, now mind you, may elicit systemic effects associated with chronic inflammation in several tissues. And essentially it will lead to injury, such as seen in the bone and brain of aged mutant mice. Now again, this tells us a lot about the aging process, but it also gives us a very fine detail of what kinds of proteins may be involved in the aging process and what may result from the aging process. Now, again, it's an animal model, but we do a lot of research in animal models because we can do things like double knockouts of genes. Because remember, a mouse is a diploid organism. So you have to remove both copies, right, in order to get a null or knockout mouse in that particular protein. And so we can't do that, obviously, in human systems. We can do it in human cells, but we want to look at a whole animal system to get a better idea of what would we then move from cellular aging or senescence to homotypic organismal aging. So that's why we use these animals in these studies. They're very important. Now, let's go back to a paper published in 2006. So again, this is what you get when you listen to authentic biochemistry. I go into the scientific literature from multiple angles. I don't just do a progression from the newest to the oldest or from one specific sub-discipline domain and stay in it for a while and then move maybe to another one and try to compare with it what's going on in different systems, say the immune system versus, say, the digestive system. No, I don't do that. I'm a biochemist, so I see everything as a biochemical system. So I freely move from one major physiological system to another, keeping in mind that there are drastic and massive differences between those different physiological domains that nevertheless have as a foundational focus biochemical mechanisms, event ontologies. So again, this whole idea about this arachidonic acid, this is what I pulled then to show you what I meant by that. That's why there's the asterisk. If you notice on the last slide, I put an asterisk with that arachidonic acid release. Remember, it's a phospholipase A2, which cleaves that bond, right? That oxygen ester bond of that fatty acid bound to that glycerol backbone. Right? So arachidonic acid participates in the reacylation, deacylation cycle of membrane phospholipids. And that's called the Lanz cycle. And it serves to keep the concentration of that particular free fatty acid in cells at a very low level. In fact, free fatty acids in general are not allowed to be maintained in cellular systems because they are toxic. So a major component of what you may have heard referred to as lipotoxicity involves free fatty acid. Remember, which is an amphipathic, long chain, hydrophobic, monocarboxylic acid, otherwise an alkane, or an alkene, depending on how many double bonds there are. Those structures, those amphipathic molecules, can be very destructive to organic structures like membranes. Hence, the fatty acids are almost always bound as oxygen esters or as sulfur or as thioesters. Some way of controlling the movement and flux of those fatty acids came very early in the evolution of cellular life. So, to manipulate the intracellular arachidonic acid level in phagocytes, this particular group used several pharmacological strategies to interfere with that cycle, that reacylation, deacylation pathway. They used inhibitors of the arachidonic acid reacylation pathway. They used a compound called thimerosal, and they used triacin C. Those block the conversion of arachidonate into arachidonyl-CoA, a thioester, which itself can form micelles at a certain critical micellar concentration, or CMC, and a CoA-independent transacylase inhibitor that blocks the movement of the arachidonate within phospholipids. Okay? Two different modes of action there. In addition, they use cells overexpressing this phospholipase A2. Again, that's the enzyme that they believe, way back in 2006, had a key role in controlling basal fatty acid deacylation, because it's a phospholipase after all, in phagocytic cells. Okay. So all of the different strategies they use resulted in an expected increase in cellular free arachidonate, but also in the induction of programmed cell death by the classical pathway, apoptosis. Okay. Moreover, this paper showed us, when used in combination with any of those aforementioned drugs affecting thioester chemistry or oxygen ester chemistry, arachidonic acid itself was able to induce apoptosis at doses as low as 10 micromolar. So just the free fatty acid could induce cellular apoptosis. Blocking cyclooxygenase or lipoxygenase, those are enzymes which act upon arachidonyl-CoA, okay, pumping molecular oxygen into that very long chain polyunsaturated omega-6 fatty acid. Slightly different mechanisms, certainly different tissue compartmentalization considerations for the two COX and LOX enzymes, that's cyclooxygenase and lipoxygenase. Now, they say when they block those enzymes, oxygenating enzymes, which normally metabolize arachidonate, there was no effect on the induction of apoptosis by arachidonate. So it wasn't to do with icosanoids, these highly reactive oxygenated fatty acids, which probably was considered the potential candidate, which is causing all of this programmed cell death. So essentially, arachidonate levels within the cells may provide an important cellular signal just by itself for the onset of apoptosis and indeed perturbations of the mechanisms controlling that arachidonate reacylation to either CoA or again back as an oxygen ester. Hence, free arachidonate availability may decisively affect cell survival. It's something they determined way back in 2006. Okay, that was in a journal of lipid research, by the way, excellent journal. All right, keep going here. So this is all leading to, so I'm getting very specific, but this is all associated with aging. Because what we find is that this enzyme seems to enhance the rate of aging in these animal systems. Okay? So the livers of the IPLA2 beta double knockout mice show an increase in apoptosis associated with an increase in inflammation. So while that double knockout mouse physically appeared normal, they exhibited in vivo susceptibility towards lipopolysaccharide, which is a very powerful antigen from gram-negative bacteria, induced liver injury via toll-like receptors, Con-A-induced autoimmune hepatitis, and dextran sodium sulfate-induced colitis. So many of the diseases that we normally think about in high morbidity during aging. Okay. So the enzyme, that phospholipase, very specific subform of it, mediates apoptotic cell clearance through the generation of lysophosphatidyl-choline, that's the other product, thus regulating the speed and directionality of monocytes, which are innate immune cells, during chemotaxis, movement of those monocytes to an inflammatory tissue site. So this defect in apoptosis involves apoptotic cell clearance removal, and that might explain the observed susceptibility of the double knockout mice towards stress-induced injury. So that now brings into focus how a monocyte macrophage cell lineage, an innate immune cell lineage, is affected by a non-calcium dependent phospholipase A2 in the membrane via arachidonic acid associated apoptosis or programmed cell death. So damaged and dying cells promote a seek and destroy signal, where a seeking signal is a lysophosfatidylcholine. Remember, that's one of the products of the phospholipase A2, lysophosfatidylcholine. Phosphatidylcholine is a very common glycerolipid in the membrane, which is coincidental with cleaved, this is now an apoptotic signaling pathway, caspase-3 mediated apoptosis. So it's it is the lysophosfatidylcholine which seems to be triggering the apoptosis. Mice deficient in the signaling, in the seeking signal, lysophosfatidylcholine, fail to remove the apoptotic cells, which become secondary necrotic because of this, and subsequently trigger the pro-inflammatory response by the immune cell lineage. You see. So you need the signal of one of the products, not arachidonate, but the lysophosfatidylcholine. So mice lacking G protein coupled receptor 132, also known as G2A-R, it's a gene, human, Well, this would be the mouse gene, but there's an analog in humans. And that is itself a lysophosphatidylcholine receptor. And these mice develop an autoimmune disease very similar to lupus. Isn't that very interesting? All because of the levels of LPC, which is again phosphatidylcholine minus its fatty acid in the 2 position on that glycerol backbone, generating a lysophosphatidylcholine. And then let alone whatever arachidonate does in functioning downstream to coordinate this response towards autoimmunity disease. There's a lot of detail you pick up here just from looking at one gene knockout, you see. So to complete this description, see, this is why I'm going into the detail here. I'm showing you how you need to understand the detail of the system in order for us to focus on what are the properties and principles of the dialectic description of the event ontology of human aging. And of course, we're using animal models when we can and when we need to. So an inappropriate cytokine release by macrophages and indeed T lymphocytes during the inflammatory response and then associated infection will become an important component in the development of autoimmune diseases and cancer. Later on in the lifespan of that, in this case, double knockout mouse. Because you've corrupted now the immune response. You corrupt the immune response, what can happen? What's likely to happen? Either a hyperinflammatory response or a hypoinflammatory response or a prevention of cleaning up of decaying, dying, programmed cell death type products. Those cells that are in the tissue, lack of removal of those, leading to a necrotic event, thus exploding a new inflammatory signaling phenotype. So in that study, these researchers asked if macrophages and lymphocytes isolated from that double knockout mouse would exhibit altered cytokine release. Because they're saying maybe this is now heralding in a systemic response, not just a cellular response to the removal of that calcium-independent PLA2 beta. So in those mice treated with pro-inflammatory anti-CD95 FASL antibody may cause exaggerated cytokine release. And that's normally what induces the cytokine release using the FASL antibody. Okay? Because FASL normally will draw down the activity of particularly T lymphocytes, right? Because that, along with the cell death signaling in T lymphocytes, shuts off T cell activity, right? Okay. That's the way this kind of work is done. So perhaps the deficiency in that phospholipase A2 could affect lymphoma incidence of a cancer-prone immune organ. And in this particular instance, we're talking about mesenteric lymph nodes, or MLNs. These are all now co-descriptive responses in association with aging by studying it in the animal model, the mouse model. Okay? And obviously, we can't do this kind of model in a human because we're talking about doing a deletion of a gene. Right? So we learn a lot from this. Doesn't mean that we know this exactly what happens in humans, but we know that there are mutations in, for example, the calcium-independent phospholipase A2. And we see these pleiotropic effects on infection, on hyperinflammatory responses because of a corruption of the monocyte macrophage lineage and the T lymphocyte lineage because of cytokine production. Now we can go back and look at the animal study and you can see, at least get a handle on how they're linked. That's the important issue to keep in mind. Okay. Now, The results of their paper, again, I went through a lot of detail here. This is old in terms of a lecture I did quite a while ago, but you see how this gives us complete in-depth insight to the whole system. So what we say is that a deficiency of this calcium-independent phospholipase A2 beta increases apoptosis in spleen, and it primes the splenocytes then for a Th1, Th17 response. Remember, the spleen itself is what? It's an immune organ, which helps mature T effector or T helper lymphocytes. So the deficiency in the phospholipase increases apoptosis in the liver, and that's associated with a suppressed cytokine release from killer cells, so-called CD8 positive cells. These are T lymphocytes. So sublethal dose of the CD95 FASL treatment, which means that's going to regulate the potentiating activation of T lymphocytes. When you do a sublethal dose of that CD95 FASL treatment, it primes mutant natural killer cells for an enhanced M1 cytokine release. Now, the natural killer cells are working with the macrophages, and M1 macrophages will generate a lot of pro-inflammatory cytokines. So the sublethal dose treatment primes the mutant liver lymphocytes then, that's what we're talking about here, the lymphocytes, for a weak increase in Th1 cytokine release. Now, the upshot of this is with aging, an increase in apoptosis in the MLN will be associated with an inherent loss of beta cell cellularity with decreased lymphoma. Now we learn a lot about insulin in association with cancer by studying one relatively under-described phospholipase A2 isoform. And once again, the detail of it had to do with the lysophosphatidylcholine binding to its receptor, which is basically a G protein mediated event, regulating the T cell responses in reference to macrophages. Remember, macrophages are also antigen presenting cells. T cells need antigen presentation. And this all linked up to a specific CD8 positive T cell lineage with natural killer cells. So see, we go way into depth to understand aging this way. But what it tells us is that we needed all this information to get from the double knockout, which we did, just to look at patterns of what goes on when you inhibit that deacylation, reacylation system. Now with the arachidonic acid moving around, that goes through phospholipids within the membrane. That's why it's called phospholipase, by the way, because these are phospholipids. PC is a phospholipid, phosphatidylcholine, right? And then all of a sudden they find out that arachidonic acid is only playing a minor role. It's the other product of that reaction, the lysophosphatidylcholine. Thus mediating and then mitigating this entire pro-inflammatory state, moving from either programmed cell death to oncogenesis in the process. Because you get a decrease in lymphoma, of course, that's an oncogenic system. So that was a detailed discussion of one particular paper, but it goes all the way down to the core. This is what we do in authentic biochemistry. Now, here's a paper published in 2013 looking at obesity and neurodegenerative diseases. Why is this significant? Neurodegenerative diseases are very common in the elderly. Alzheimer's disease, Parkinson's disease, prefrontal dementia are three, Lewy body dementia. Right. So here we go. Aging in association with obesity, which is then linked to inflammation, reactive oxygen stress or oxidation stress, oxidative stress, growth factor loss, and then a corruption of the neuroendocrine system because of lack of positive signaling, all of that, and also with associated proteinopathies because of a corruption of glycosylated protein biosynthesis in the endoplasmic reticulum because of an ER stress response, will lead to neuronal loss, and all of that will give us age-related neurodegenerative disease. And when we get age-related neurodegenerative disease with aging and all these other possible pathophynotypes, which are linked to it, as I just pointed out, I hope, you get mood change, memory loss, movement dysfunction, the classical phenotypes of the elderly. Okay. That allowed me to summarize all the previous work there into that slide. Now, this gives us a lot of information. I'm going to just move myself out of the way here so I can discuss it. This slide shows us senescence-mediated neurodegeneration. It's a 2019 paper published in Frontiers Neurosci. And it shows us that cellular senescence is induced by multiple stimuli, such as ionizing radiation, reactive oxygen, if a person is under chemotherapy, but then also obesity, the disease obesity, and then stress, which can be quite pleiotropic in the human system. The cells involved include oligodendrocytes, microglia, astrocytes, endothelial cells, and particularly the pericyte, which is a kind of an endothelial cell. Cellular senescence, as I told you about 20 minutes ago, is linked to certain cell cycle protein activities, the P16 Inc4A, P21 CYP1 WAF1, the SA beta gal, which has been used as a biomarker for aging, DNA damage, and of course, the increase in heterochromatin foci. Heterochromatin is a collapse of chromatin. That means a decrease in the overall generic gene expression at the level of transcription. So these parasites, which basically are the cells which comprise the blood-brain barrier, you get increased permeability with all the cellular senescence in association with those cell cycle dependent proteins being altered in their activity and then limiting to DNA damage and heterochromatin foci will increase blood-brain barrier permeability. At the same time, you're getting SASP increases because of senescence. This can lead to a problem with lipid manufacture, that is biosynthesis, particularly sphingomyelin. And SASP and SAS phenotype is going to limit myelination. And we know that myelinated neuronal bundles are necessary for quick activity in the brain because of saltation processes of the myelination. Myelination means sphingomyelin coating on these particular neurobundles. What you then get is a protein called NFT depositing. Also, neuronal degeneration occurs, brain shrinkage occurs, and loss of cognitive function. All of that is neurodegeneration. So you have senescence-mediated neurodegeneration being affected within all those different innate immune cell lineages, the microglia, the astrocyte, the oligodendrocyte, all can act as immune cells in the CNS, particularly the microglia, all functioning by causing leaky parasites, which are the cells of the blood-brain barrier, and then association with changes in the genes and the expression for the cell cycle, P16, Inc4A, etc. So you get senescence of microglia and the astrocytes. That results in an inflammatory response and the loss of trophic support. That means the oligodendrocyte senescence reduces myelin synthesis, sphingomyelin biosynthesis, which would ultimately affect neurotransmission and the blood-brain barrier can become compromised by endothelial cell senescence, such as the parasite. All of these changes have an impact on the integrity, viability, and activity of neurons, and then, of course, downstream from that, brain function. This, again, is what you get from the paper published in Frontiers in Neuroscience. Okay? So hopefully that was a good way to synthesize all this information that we've been leading into, as we go. Now, what about the heart? It's a paper published in the Journal of Cardiology, again, a few years ago, 2019. So let's take a look at that. And I'll pull myself back up here so you can see me again. Senescence dictates cell division exhaustion, and we call that cellular senescence. Remember, we've already introduced that. And that phenotypically presents as flattened, enlarged, and irreversibly re-altered at a morphological level cells. Therefore, they are unable to reach the next cell division. Proteins involved include P53, P21, the cell cycle protein, again, P16, Inc4A, the kinase cascade proteins, P38 MAP kinase, as well as the gamma H2AX, telomere attrition because of telomerase activity decreasing, right? We talked about this. Enhanced signals such as the SA beta gal, which is a biomarker. And all of this is associated with senescent cells, right? So you get a lack of the necessary contribution, which would be given by non-senescent cells. So once you start to senesce, all of those functions start to become limiting, right? So of course, the SASP phenotype is characterized by senescent cells secreting pro-inflammatory cytokines. And so you get this pathophysiology in aging. And it's dependent on the aging process because of the senescence. And it correlates sometimes with morbidity of heart failure. Finally, we're getting to the heart system, right? And that's linked to atherosclerosis, which is a lipid-mediated response having to do with the production of reactive oxygen, the induction of certain eicosanoids, and then the activation of innate and then acquired immune, cellular, and humoral responses, leading to plaque formation because of the integration of macrophages into the cardiac muscular cellular system. This generates foam cells, which will then decrease heart output, generating ultimately heart failure. So, of course, SASP is a chronic, yet it's a sterile, which means it doesn't induce a leakage of the cells because these cells are active, they're secreting, but they're not exposing to necrotic cellular degradation. These senescent cells are not necrotic, right? So not leaking content. So we say it's a chronic sterile inflammatory associative tissue remodeling. That's basically what the SASP is. And it happens right in the cardiovascular muscle system in association with increasing potentiation of atherosclerosis. Okay? So senolysis is an incompletely characterized specific elimination of senescent cells. So then, with papers like this being published, cardiologists are saying, hey, can we find ways to promote senolysis? Basically causing SASP phenotype to disappear by causing those senescent cells to apoptose. You see? That's called senolysis. You got to keep track of these terms here. So cytogenic and pharmacological models of senolysis have been touted as reverse aging phenotypes, as you might guess. If you can get rid of the senescing cells, then you're not going to promote this aging phenotype, such as in the cardiac muscle or in the central nervous system we just described. You know, the blood-brain barrier, the parasites, and all that. Right? Two very, very important foci for the aging human. Right? And so they argue maybe you can get an adaptive departure from a pathophysiological response like that, which is typically obtained by an aging-related morbidity. See, that's the argument here. Senolysis research promotes a theory of reduced cardiovascular disease, and it suggests that couples with no increased risk of tumorigenesis. Now, that is a tremendous ask of a system. If you are promoting programmed cell death, and that's believed to then enhance the more immature state of the cellular system because you're getting rid of more senescent cells, that the senescent cells have nothing to do with oncogenesis, but we just told you they do. In fact, the senescent cells suppress tumorigenesis. So to suggest that getting rid of senescent cells won't affect tumorigenesis seems to be a big ask, you see. So physiological, nutritional, pharmacological therapies prompting senolysis, according to this paper's discussion, could promote healthy cardiovascular aging. Now, I can tell you this has been studied, and I can tell you what you already can probably guess. Promoting senolysis does not go without negative feedback event. Promoting it will remove some of the control over the oncogenic responses. That the SASP system, the phenotype from senescent cells, secreting, for example, pro-inflammatory cytokines or growth factors or chemokines, which can control massive cell division through those cell cycle protein mutations, which I told you can occur, the P16-INK4A, for example, that would all be lost if you promoted beyond the range that was physiologically promotive a senolysis phenotype because of using pharmacological agents, etc. And they're suggesting a nutritional modification of the diet, basically. So what I'm telling you is that when you push on one side, you get the other side pushing back. So you might be able to control the senescent response, which looks like it could be good for the cardiac muscle because of this lack of foam cell integration and all this. At the same time, you can promote oncogenesis, which is another leading cause of death in elderly. All right, so paper here talks about this particular protein, INK, and it's the ATTAC mice, a specific phenotype. And those mice were generated. I'll get into this detail again because this is what we're doing here. We're getting into detail, and then we're moving out of the detail and talking generically, or conceptually, I should say. So those mice were generated to enable researchers to produce an inducible elimination of that cell cycle protein, the P16-INK4A. Yeah. And they do it by an administration of a compound which will cause this elimination. So it's an inducible deletion, a model. So in a prematurely aging mouse model, the elimination of the P16-INK4A positive senescent cells, remember these are going to be positive for that cell cycle arrest enzyme, leads to an inhibition of aging phenotype in white adipose tissue and in the heart and in the kidney. Okay. Now, elimination of those P16-INK4A positive cells in the murine model didn't appear to change heart rate, left ventricular LV mass, thickness and diameter, ejection fraction, and fractional shortening compared to the control. Those are all monitoring heart output. Left ventricular mass is used to evaluate the risk and the prognosis of patients with heart diseases and of course, associated heart failure. And it's used as a diagnostic for significant risk to patients' life and health. So here you got it. The normal range here, right, for left ventricular mass, which would otherwise, when it increases, is called hypertrophy. In women, this is in grams per meter squared. The normal is between 43 and 95 grams per meter squared. Mildly enlarged, but abnormal, 96 to 108. Moderately enlarged, and severely enlarged is anything greater than 122 gram per meter squared. Men, the numbers are a little bit higher. 49 to 115 is normal. 131 is a max for mildly. 148 is a max for moderately enlarged, and anything greater than 149 is severely enlarged. Right? This again is hypertrophy. Hypertrophy is associated with cardiovascular disease, right? So an accumulation of these P16-4A positive cells is associated with pathogenic cardiac aging. That's what they've discovered. A transgenic mouse that kills those cell cycle protein P16-4A positive cells upon administration of this another inducible system using ganciclovir. This is using negative selection I talked about before. I won't go into it now. Leads to a regression of atherosclerotic plaques in low-density lipoprotein receptor deficient lines. So that's an artificial induction of atherosclerosis. It suggests an association with cholesterol oxidation, which is associated with foam cell formation, and or triacylglycerol accumulation. Okay, because you're working against the LDL fraction, which is enriched with triacylglycerol. Okay. So the clinical manifestation associated with immunosenescence. So that's one important thing to consider. You can decrease the senility, the senile function of the cells, decrease the amount of senescent phenotype, enhance heart muscle activity in the aging mouse model. At the same time, though, you can be altering possible oncogenic events. Okay. So, paper published now in 2020 in BMC Cancer told me the following. Clinical manifestations associated with immunosenescence, such as SASP, in the elderly included decreased ability to control infection. Remember this, talked about it. Poor response to vaccination because of the lack of B cell responses and T cell associated activation of plasma cells, and an increment in the risk of developing cancer. Okay. Several immunological alterations have been described with aging, including changes in T and B lymphocyte subpopulations and the NK cells. Those are CD8 positive T cells. So the B lymphocyte compartment also shows a decrease in naive B cells with a reciprocal increase in the memory B cells in immunosenescence. More memory cells, less naive cells, which means less clonal propagation of specific IgG and immunoglobulin series, antibody production, thus lacking the focus of a plastic and elastic response linked to new pathogen invasion, which is normally what's required from naive B cells, because now you have more mature memory cells, which are specific to previously encountered antigens. So the B lymphocyte compartment, again, shows a decrease in naive B cells. Aging is characterized by a decrease in circulating antibody, hence the problem with vaccinations in the elderly. At the functional level, natural killer cells exhibit decreased cytotoxicity. That's because of changes in metabolism in those cells. And natural killer T cells reduced migratory capacity. They can't move very well and they can't kill very well. Okay. So the P16NK4A, which is again a cell cycle regulating polypeptide, and also an inhibitor of cyclin-dependent kinases 4 and 6, normally healthy people will play an important role in cellular aging and premature senescence. And in fact, it's recognized as an aging biomarker. So you want premature senescence, but you want age relative senescence, right? Age appropriate senescence. So it looks like P16-INK4A expression in peripheral blood T lymphocytes is described as sharply increased with chronological age. Isn't that interesting? And it contributes to an age-induced functional decline of certain self-renewing compartments, such as lymph glands. Yeah. So oncogenesis eventuates a genetic mutation to an epigenetic gene expression mechanism, with molecular signatures either inappropriately proscribed, incorrectly prescribed upon writing as dangerous and deleterious, or the process of erasure of those epigenetic signatures. That leaves a corrupted chromatin. That's what oncogenesis does. Tumor cells obtain proliferative autonomy, autophagous self-maintenance, in growth and signaling, neovascularization for nutrient and oxygen supply, and a resistance to anti-proliferative and apoptotic stimuli. In resting cells, the cell cycle is strictly managed by a set of regulatory proteins that control the variations in cell cycle checkpoints, such as the P16-INK4A and P53, and the RB protein, and that will become dysfunctional during the early transforming stage of the tumorigenesis response. And that's via an unregulated dismantling of those same tumor suppressor genes, which we're arguing also controls senescence, right? Particularly the whole description in the cardiac muscle and in cardiovascular disease, right, that we just went through. A senolytic response, right? So when a biochemical event is de-repressed, it's brought back to register. However, when it's brought back to register, the valence is lost. Like where is it going to be weighted in the dialectic of cell activation and agency? See? In effect, this dismantling is a repression of tumor suppressor gene transcription and or translation, post-translational modification of those proteins, and then, of course, all of that leading to their final agency, or that is their biopotency, biological activity. So the loss of gene suppressor activity, which we were just talking about with the P16-INK4A, which looks so good for decreasing cardiovascular disease, right? For the reasons we pointed out. The loss of gene suppressor activity can result from any number of mechanisms: deletion, inactivating mutations, epigenetic silencing, incoherent post-translational modifications, such as phosphorylations, acetylations, ubiquitinations, propioninations, malinations, methylations. And not only that, but also glycosylation, acylation, prenylation, and phosphorylation. That can also alter aggregation of polypeptides in the cell cycle, associated with this repressor activity. You also get lipid-mediated transport aberrations because of this. And that means lipid RAS from sphingomyelinase-mediated ceramide synthesis will then become dysfunctional. The typical biomedical result is oncogenesis, which of course is transformative, and ultimately tumorigenesis, which then becomes committed to cancer. Metastasis, which means the mobilization and transactional activity of the cancerous state, and either death of the cells or repair to a healthy state. And we know which direction normally goes is towards death and more mutation and more cellular damage, right? So the progression of the mammalian cell cycle from G1 to M1, mitosis, is of course regulated by those cyclins, such as the 16-4A. And of course, with all of their cogent catalytic subunits, and that whole system itself are known as the cyclin-dependent kinase, or CDKs. We talked a lot about this over the months. So family of cyclin CDK inhibitor proteins called CDIs, which bind and inactivate the kinases, include those proteins, those so-called repressor proteins. That's why they're repressors to cell cycle. 16-4A, P21 CYP1, P27 KIP1, you remember these now. And there are other proteins involved, as you can see there, many more. Those proteins potentially act as tumor suppressors, and their inactivation will then correspond to human now carcinogenesis. So while you look like you can alleviate the age-associated cardiac system decline at the same time, because you're corrupting what normally is occurring with cell cycle mediated responses, now you're going to increase the potentiation of carcinogenesis. So whenever you try to change what is the normal lineage-specific pathway of aging unto death, you're going to tip the balance between apoptosis, or just basically cellular death. That could include, of course, necrosis and pyroptosis and inflammatory responses, loss of function because of loss of cellular activity, but still a robust until high morbidity of that senescence-associated secretory phenotype. That on one end of it, and on the other end, is the potential for massive cell division in cancer. And aren't those the two main causes of death in humans? They are cardiovascular disease and cancer. So there's an equipoise which is maintained until such time that there is a dysregulation of the functional domains at the biochemical level that regulate those pleiotropic, fundamental, cellular, progressive phenotypes. And any interruption or alteration of those will lead to further corruption of the system, which could either cause massive cell death or massive cell division. Yeah. So I hope you get that the point I'm trying to make there, because it's very significant relative to what phenotype we can hope to obtain by, for example, pharmacological means of the attempts to prolong the life cycle, right? Too many multiple domains, iterative multiple domains of regulation that could otherwise become corrupted by massive gene expression changes, such as using CRISPR to alter the expression of genes, or putting in new genes, or removing old ones that appear to be deleterious to living longer. All that's going to corrupt all this fine-tuned, highly calibrated progression of the living system towards morbidity and death. It could hasten the process or alter any one of the functions that are otherwise absolutely necessary for co-occurring for a healthy lifespan, at least until the end. So I'm going to leave you with that now. Told you that this was not going to be the last time you see me, because this lecture would be way too long. We're going to continue right along, though, with this last lecture You're just going to be cut up into multiple, I guess, about 1 hour segments. This was the beginning of the end, I promise you. And I would continue on and on and on. But I think it's better to stop here, digest what we've talked about, and then we'll pick up right at this point next time here on Authentic Biochemistry. So, again, this is Dr. Dan Guerra coming to you on the 4th of December. Now it's nighttime here in the Pacific Northwest. Wishing you a very pleasant weekend. And we'll come back to you very soon with the second installment of the last lecture on the aging phenotype, or what I'm calling the sickness unto death. Okay? So, signing off, Dr. Dan Guerra. Bye for now.